Welcome back, everyone. In our previous session, we have explored the concept of carbohydrates and lipids. Today, we shift our focus to the third and the most versatile macromolecule, that is, proteins. My dear students, proteins are not just muscle builders, they are the fundamental machinery of life. And in this lecture, we are going to break down the chemical composition of proteins, the complex processes of protein synthesis. Then we shall discuss about protein structure and its functions. And the most important part is the levels of protein architecture. And in this lecture, we are also going to discuss briefly about enzymes and antibodies and how proteins are denatured. First of all, make sure you stay tuned until the very end because we will be solving a set of high yield practice questions that directly mirror what you will see in your final exams. So, if you want to master biology and stay ahead of the curve, join our learning community at Dr. Wasif Academy. Hit that subscribe button and turn on the bell icon so you never miss a breakthrough lecture. Okay, let's dive in. Now we come to the most versatile molecule in all of biology, which is protein. If carbohydrates and lipids are the fuel and the walls of a cell, then proteins are the machines. They do the actual heavy lifting from moving your muscles to digesting your food. So proteins are large polymers made of smaller monomers, and these monomers are called amino acids. But check out the chemistry here. It's a bit different. Like everything else, proteins also have carbon, hydrogen, and oxygen, but the absolute identifier, as we discussed in the previous slide, the absolute identifier for a protein is nitrogen. Okay, I have written down it here. All proteins must contain nitrogen, and many also contain sulfur. Think of sulfur like the industrial glue or bridges which lock a protein into a very specific 3D shape. And without those sulfur bridges, the protein would just be a limp string and would not be able to do this job. As a takeaway tip for your exam, which is a classic exam trick, if you're looking at a diagram and you see the letter N for nitrogen or S for sulfur, you're not looking at a sugar or a fat. You are looking at a protein. And finally, for the lab, how do we find them? We shall discuss later, okay, under the heading of Burette test. So I am skipping this slide here, this point from here. Now let's look at how proteins are actually built. I like to think of amino acids as the alphabet of life. There are 20 different types of amino acids. Just like you can rearrange the letters of the alphabet to make thousands of different words, this cell rearranges these 20 amino acids to build every protein in your body. But here is the most important part, that is the sequence. Okay, the sequence or the arrangement of these amino acids that actually matters. Once this long chain, which we all call a polypeptide, is built, it does not just stay as a string. It folds, it twists and curls into a very specific 3D shape. Think of it like a key. If you change even one tooth on a key, it would not fit the lock anymore. It is the same with proteins. If you change just one amino acids in that long chain, the whole 3D shape might change. In biology, if the shape changes, the protein can no longer do its job. If you remember only one thing today, let it be the golden rule. That is simple equals shape equals function. Enzymes have an active site shaped perfectly for the substrate, but antibodies are shaped to fit a specific germ or pathogen. So hemoglobin is shaped specifically to cradle oxygen. So if an exam question asks why a protein stops working, maybe because the temperature got too high, the magic word is denatured. It means the 3D shape has been destroyed due to high temperature. So the lock is broken and it can no longer fit its target. Now let's categorize these incredible molecules. You can think of proteins as being either the scaffolding or of the body or the tooth of the cell. First, we have structural proteins. These are the framework. Inside your cells, they sit in the membrane acting like specialized gates or transport channels. On a larger scale, they build your body. Collagen gives your skin and bones their incredible strength, while keratin makes your hair and nails tough and waterproof. Then we have the biological tools, the enzymes. Okay, in previous slide, we discussed about lock and key model. Okay, I think that you could not understand that point in the previous slide. Now in this slide you can understand that to some extent, but on later slides that will be in detail. So enzymes are biological catalysts. Their mission is simple: speed up chemical reactions without being used up. Without enzymes, the digestion of your breakfast or the respiration in your cells would happen so slowly that you simply could not stay alive. If you look at this picture, okay, this is the active site of a substrate. Okay, this is the enzyme active site. Okay, this is the substrate, and this substrate fits into this enzyme. This is the key and lock model. Okay, lock and key model. Then it can easily perform its function, and after the completion of the function, the products are produced, and this enzyme will be eventually draws itself off. So when you are drying a cell membrane, remember this: the lipids make the wall, but the proteins are the gatekeepers. You must draw them as intrinsic proteins which span the membrane. And here is a tip for those define and exam questions. Even if you are stuck, always write down the word biological catalyst. Enzymes are biological catalyst, and mention that they lower the activation energy. Those two phrases are almost always on the marks scheme. We have reached the big picture. To truly master this unit, you need to be able to look at any molecule and immediately identify its group, its ingredients, and its job. Look at this comparison table one more time. Notice the pattern. Carbohydrates and lipids are your pure energy and structure molecules built only from carbon, hydrogen, and oxygen. But the moment you see nitrogen or sulfur, you know you have moved into the world of proteins, the molecules which actually do the work. Think of the logic like this: if an exam question asks you about fuel, whether it is the fast fuel of glucose or the long-term storage of a lipid, your mind should go to C, H, and O, carbon, hydrogen, and oxygen. But if the question asks about doing a job like speeding up a reaction with an enzyme, carrying oxygen with hemoglobin, or defending the body with antibodies, you are talking about proteins. This is my final tip for this unit, the nitrogen test. Okay, this is just a brief note here that in your multiple MCQs paper, multiple choice question paper, not waste time overthinking complex diagrams. Hunt for that. And if you see nitrogen, it is a protein. If you only see C, H, and O, and there is no nut, no N alphabet, no S alphabet, then you have narrowed it down to a sugar or a fat. And remember, you should remember, structure dictates function. Whether it is the solubility of glucose or the 3D shape of an enzyme, it is the chemistry that makes life possible. Now, the next topic is protein architecture. We have understood that proteins are made of amino acids, but a protein's power does not just come from its ingredients. It comes from its architecture. Think of an amino acid chain like a piece of wire on its own. It is just a string, but if you bend it perfectly, it becomes a functional tool. Because there are 20 different amino acids, the number of ways you can link them is nearly infinite. This specific sequence acts like a set of instructions. It dictates exactly where the chain will bend and where it will fold. As the chain grows, chemical bonds like sulfur to sulfur bridges form between amino acids that might be very far apart in the line. These bonds pull and lock the protein into a very specific, stable 3D shape. This folding creates the pockets or active surfaces, which allow the protein to recognize and interact with only one specific molecule. This is about specificity, okay, which just I just told you. And this is how you answer question about mutations. If an examiner asks what happens if the DNA changes, so follow this logic. A change in DNA changes the sequence of amino acids. How that comes, this we shall study in detail under the content of DNA. Okay, but he's just you can say that just a introduction. Okay, a change in DNA changes the sequence of amino acids, and this changes where the bonds form. Therefore, the protein folds into a different 3D shape. The active site is lost and the protein can no longer function. So to get higher marks, always link those three keywords: sequence, folding, and 3D shape. Or you can say that as specificity. We have seen how proteins fold, but now let's see that folding in action, the lock and key principle. The lock and key principle is the best way to understand how a protein's 3D shape allows it to do highly specific jobs. First of all, look at enzymes. The protein folds to create that specifically shaped pocket we call the active site. We call the This is the active site. It is built to have a complementary fit with a specific substrate. Okay, this is the substrate. Think of the precision here. If the temperature or pH changes very even slightly, the protein shape shifts. The key will no longer fit the lock and the life-sustaining reaction stops immediately. But proteins do not just build and break things, they also defend you. Antibodies are proteins produced by your lymphocytes. They have arms with binding sites which perfectly match the antigen on a pathogen surface like a virus or bacteria. By looking onto the enemy, the antibody flag it so your other immune cells know exactly what to destroy. I cannot stress this enough here. Okay, use the C word. Never say the substrate and enzymes are the same shape. They have complementary, like a hand and a glove. Examiners hunt for this specific word. This also explains why a flu vaccine does not stop a common cold. Your antibodies are like custom-made keys. Their binding sites only fit the unique antigen shape of the specific virus. They were built for the specific purpose. If there is no fit, then there is no protection. We have seen how important that 3D shape is. But now we have to talk about how easily it can be destroyed. Because protein shapes are held together by delicate bonds and cross linkages, they are incredibly sensitive to their environment, especially heat and pH. Think about what happens when you turn up the heat. As the temperature rises, the molecules start to vibrate violently with kinetic energy. Eventually, what will happen? That vibration becomes too much, and the delicate bonds holding the protein together will simply snap. The protein unravels and loses its functional shape. This process is called denaturation. Once this happens, the active site of an enzyme or the binding site of an antibody is gone. There is a clear damage. It is like melting a key. It does not matter how much you try to shove it onto the lock. You try to fit that into the lock. It simply would not fit anymore. And here is the kicker. Denaturation is irreversible. Denaturation is irreversible. To remember this, just think of a frying egg. When the egg is raw, the proteins are clear and runny. But once you apply heat, they denature, tangle together, and turn into that solid white. You cannot unfry an egg, and you cannot undenature an enzyme by cooking it back down. So what is the critical phase here? If you want good marks on your exam, you must use this exact sentence. The active site has changed shape and is no longer complementary to the substrate. So memorize that phrase. It is your ticket to a grade A. As we have seen how heat and pH can unravel a protein, can denature a protein, but what does that actually look like for a living organism? When proteins fail, the consequences are not just a minor glitch, they are fatal. Think of it as a factory where every single machine suddenly melts. The building might still be standing, but the work has stopped. First, we see a metabolic collapse. Because enzymes are denatured, essential reactions like aerobic respiration and DNA replication simply stop. The cell can no longer produce ATP. ATP is what is ATP? That is the energy currency. Okay, energy currency of life, and it loses the ability to repair itself. Next, you get structural failure. Remember, those protein gatekeepers in the cell membrane we talked about, when they denature, the membrane loses its integrity. The gates fall off their hinges, and the cell can no longer control what comes in or what goes out. What are critical challenges here? So critical organelles like your mitochondria and ribosome literally fall apart. And finally, this leads to cell death. The cell loses its ability to maintain homeostasis or internal balance. What is homeostasis? That is internal balance. Once the proteins are unfried and tangled, the cell is no longer a functional unit of life. It is just a disorganized mess of chemicals. This is exactly why a high fever or extreme dehydration is so dangerous. It is a direct attack on the machinery which keeps you alive. Now we all have understood that a high fever feels terrible. We all know that high fever feels terrible, but biologically it is a race against time. Your body normally sits at approximately 37 degrees Celsius. This is not a random number. It is the optimum temperature where your enzymes are at their absolute peak performance. But let's look at the danger zone. If a fever pushes your temperature towards 40 degrees Celsius or 42 degrees Celsius, the kinetic energy in your cells become too high. The delicate hydrogen bonds and sulfur bridges, which hold your brain and organ proteins together, begin to vibrate and snap. At this point, you are quite literally cooking your internal machinery. As these proteins denature, vital signals in the heart and brain stop. This is why a sustained high fever can lead to organ failure, and the machinery of life is being dismantled by the very heat which is meant to fight the infection. So my dear students, you must be able to describe the optimum graph. Okay, from 0 to 37 degrees Celsius, the rate of reaction increases because molecules move faster and collide more. This is basic kinetic theory. The peak at 37 degrees Celsius is the optimum. Above 40 degrees Celsius, the rate does not just slow down, it crashes. Why? Because the enzyme is denaturing. A final warning for your exam is here. You can say final warning or for a clue here. Never say an enzyme denatures when this is cold. Okay, I repeat it. Never say an enzyme denatures when it is cold. At 0 degrees Celsius, it is just inactive. It does not denature. Okay, it is just inactive or moving too slowly to work. So denaturation is a one-way street caused only by high heat or extreme pH. Here is a question for you to wrap up. Let's look at this question, which that appears in almost every single exam, or which might appear in any exam. Why is the specific 3D shape of a protein so important for an enzyme? To answer this question, we use the lock and key hypothesis, or you can say lock and key model. Imagine every enzyme has a uniquely shaped pocket called the active site. For a reaction to happen, a specific molecule, the substrate, must fit into that pocket. I have explained that in detail with as a pictorial diagram. Okay, you can move to that slide after understanding this. The shapes must be complementary. Okay, means the shape of an enzyme and a substrate means a key and a lock. They must be complementary. Think of it exactly like a key and a lock. If the key does not match the internal shape of the lock, nothing will happen. This is why enzymes are so specific. A protease enzyme is shaped perfectly to break down proteins, but it will have absolutely no effect on starch because the shapes simply do not match. It is the wrong key for the lock. And this leads to the concept of denaturation. If you turn up the heat or change the pH too much, the bonds holding the protein together will snap. The protein denatures and its 3D shape changes. Its specific shape gets changed. Okay. Now the active site is no longer complementary to the substrate. The key no longer fits the lock, and the reaction stops. And here's a critical tip for you. I have seen this mistake many times. So you should never say the enzyme dies or is killed. Enzymes are just chemicals; they are not alive. So using the word die or kill is not it does not fit here. Always use the word denatured to describe this change in shape. Use that word, and you will secure good marks. So I am concluding this video lecture here. Okay, tomorrow we shall start with the remaining of the chapter. So stay subscribed, and if you have any concern, any question about today lecture, just you can send me an email or you can comment in the comment box. Then I shall definitely respond to that particular comment. So see you tomorrow with a new topic. Goodbye.